Assalamualaikum and salam Yatim di hatiku. Today is such a beautiful day. Now we're gonna present about chromatography of Kale under chemistry 256. We are from F4AS 120412 and this will be present for our beloved lecturer Miss Nazwa John and fellow friends. Before we go further ado, let's meet our team members. Now we will start with Nur Alya Balqis binti Shamsudin, Balqis Nur Aqilah binti Mohmad Nur Farisha Humaira binti Azwira, Yasmin Hanum binti Mohmad Yatim, and I am Nurul Ain binti Razali. Before we go further ado, this is the content of chromatography of Kale. The first one is introduction, followed by theory, methodology, results, conclusion, and reference.
So here is the introduction of chromatography. Chromatography is a general name method compound in mixture physically separate between two phases. Next, paper chromatography is technique for separate dissolved chemical substances different rate across sheet of paper. Moreover, leaf chromatography allow us to see the colorful pigment that hidden inside. Why kale leaf? It is because kale leaf contains several pigments contribute their color and their nutritional value. First one is chlorophyll. Chlorophyll is the primary pigment giving their green color chlorophyll essential for photosynthesis, allow plant absorb a sunlight. Next is a carotenoid. Carotenoid contribute yellow and orange shoot kale leaf known for the antioxidant properties. Moreover, lutein is important for our eyes health. Next, anthocyanin. We can describe it as red or purple kale contribute red, purple or blue. This pigment also have antioxidant properties.
Chromatography are divided into two phases. First, stationary phase. The stationary phase is the phase that stays fixed inside the column or on the support medium. It can be a solid or a liquid supported on a solid. Its primary role is to interact with the components of the mixture being separated. This interaction causes different substances to move at different speeds, resulting in their separation. There are two types of stationary phase. First, solid stationary phase, used in adsorption chromatography such as silica gel. Next is liquid stationary phase, used in partition chromatography such as liquid coated on a solid support in HPLC. The choice of stationary phase depends on the nature of the substances to be separated. It can have different properties such as polarity, particle size, and surface area to facilitate effective separation. The other phase of chromatography is mobile phase. The mobile phase is the phase that moves through or over the stationary phase. It can be a liquid or a gas. Its primary role is to carry the sample mixture through the stationary phase. The mobile phase interacts with the sample components, helping to transport them along the stationary phase. There are two types of mobile phase. First, liquid mobile phase uses in liquid chromatography such as HPLC and TLC. The other one is gas mobile phase, used in gas chromatography such as helium or nitrogen. The mobile phase is chosen based on its ability to dissolve the sample and interact with the stationary phase. Factors such as polarity, viscosity, and boiling point are considered to optimize the separation process.
Now we go for methodology chromatography of Kale. The materials are chromatography paper, stationary phase, chromatography chamber, a 400 ml or larger beaker with aluminium foil as cover, ruler, pencil, mortar and pestle, acetone, chromatography solvent mixture, 9 parts petroleum ether to 1 part acetone, mobile phase, kale leaves, sand, and magnesium sulfate anhydrous. Now this is the procedure. Firstly, break up one of the leaf into little pieces and place it in a mortar along with 2 grams of anhydrous magnesium sulfate and 6 grams of sand as this helps with grinding up the leaf. Then add a few drops of acetone. Next, use the pestle of mortar to grind up the leaf and to release the pigments. Thirdly, draw a line on the chromatography paper about 3 cm up from the bottom of the paper. Fourth, create a concentrated spot of pigment on the line. Fifth, pour running solvent 9 parts petroleum ether to 1 part acetone into the container for the chromatograph. Ensure it does not go above the pencil line on the paper. 6. Place the chromatography paper gently in the container and allow the solvent to begin to move out the paper, carrying the different pigment with it. Oh, this is a note for you. It is important to keep an eye on the solvent front because the pigment will stop at different distance relative to how far the solvent front goes. 7. When the solvent front has stopped moving, remove the paper. 8. With a pencil, mark the edge of the solvent front and measure the distance from the point of original pencil line that has been drawn on the chromatography paper. Lastly, mark the front edge travelled by each pigment and measure how far each pigment moves from the point of origin pencil line that has been drawn. In order to identify the pigments, firstly, you need to calculate RF value for each pigment that you found on chromatogram. So, RF value equals to distance moved by the pigment divided by distance moved by the solvent front.
Now let's move to the colorful world of kale leaf pigments through chromatography. Kale, renowned for its nutritional benefits, showcase a beautiful array of colors including green from chlorophyll, yellow from carotenoids, and sometimes even red or purple hues from the anthocyanins. In chromatography, we utilize a technique that separates mixtures into their individual components based on their affinity to a solvent. For kale leaf, we employ a solvent mixture containing hexane, acetone, and ethanol in varying concentrations. These solvents interact differently with the pigments present in the kale leaf extract, causing them to separate and form distinct bands or spots on the chromatography paper. To quantify this separation, we use a parameter known as the retention factor or RF. The RF value for each pigment is determined using the formula RF equal by distance pigment travels divided by distance solvent travels. Here, the distance the pigment travels represent how far its band moves up to the chromatography paper from the original spot, while the distance the solvent travels is measured from the original spot to the solvent front. Now, we look onto the result from experiment with kale leaf using chromatography technique. Firstly, acetone used to extract chlorophylls and carotenoids from kale leaf because these pigments dissolve well in acetone and are less soluble in water. After shaking and allowing it to settle, a layer rich in chlorophylls and carotenoids collected using hexane, which separates them based on their different solubilities. In the second part, using column chromatography, yellow carotenoids appeared in fraction 2, fraction 3 gave colorless, and fraction 4 resulted in green with chlorophylls. In TLC plate analysis, carotenoids traveled further because they are less polar, whereas chlorophylls, being less soluble in non-polar solvents, move less on the plate.
Chromatography proved to be a reliable and straightforward method for revealing the diverse array of chemicals present in kale leaf. These include vitamins such as vitamin C and vitamin K, minerals like calcium and potassium, pigments such as chlorophyll and carotenoids, and phytochemicals like flavonoids and glucosinolates. Chromatography can help quantify antioxidant compounds present in kale, which play a key role in combating oxidative stress and promoting health. By identifying and quantifying specific compound, chromatography can assess the quality and freshness of kale sample, aiding in agricultural and food science research. Understanding the chemical composition of kale through chromatography can contribute to knowledge about its potential health-promoting properties, such as its role in reducing inflammation, supporting cardiovascular health, and possibly reducing the risk of certain diseases. Findings from chromatography studies of kale can inform further research on dietary guidelines, functional food, and nutritional intervention aimed at improving public health.